much for the kind invitation to talk in this exciting symposium this afternoon about cross-species neuroimaging and human brain evolution. To understand the human brain and human mind, how we process information, how we interact with each other, how we interact with the world around us, for this we have to know the history of our species and the selective and environmental pressures that shaped our brain and those of our ancestors. Fossil studies are a prime method of studying these adaptations. Cranial capacity of modern-day humans has an expanded These paleontological records provide, however, little information about soft traits. Things like brain function and cognition do not fossilize. Another way of studying the history of our species and brain evolution is thus to compare the differences and similarities among living species and compare their cognitive processes and their neuroanatomical features that underlie them. Now, this analysis can be performed across a range of species, for example, comparing brain volume across a wide range of mammalian species or a wide range of primates, but also across and between a small set of closely related species. Now, of particular interest here is a comparison between humans and chimpanzees, as we are evolutionarily very close, with humans and chimpanzees together with bonobos sharing a common ancestor an estimated 6 to 7 million years ago. Now, the human brain is around 3 to 4 times larger than that of the chimpanzee, and the chimpanzee brain, interestingly, has an estimated equal size of brain of those of our common ancestor, as derived from these fossil studies. Now, comparative studies have suggested a large expansion of, in particular, the frontal cortex, together with a potential selective expansion of higher-order cognitive systems like the default network and parietal frontal executive networks. A former postdoc in my lab, Jongbin Wei, has performed this interesting study where we found that hard genes, human accelerated genes, are particularly highly expressed in these expanded cortical areas and systems. Now, of particular interest in this context is, of course, the field of comparative connectomics. Comparative connectomics has the goal to study commonalities and differences in connectome structure across species and how they relate to behavior. Now, graph theory and network neuroscience may provide a particularly useful toolkit for such studies. As you know, in connectomics, the brain can be described and studied as this mathematical graph, from which we then derive graph theoretical features and link these features to cognition and behavior. Now, in a comprehensive literature study a couple of years ago, Olaf Sporns and I compared the connectome organization of a wide range of species. And two central common themes of connectome organization seem to emerge. First, across all species, the connectomes show a clear modular organization, indicative of the notion of the majority of connectivity to be relatively short range in connectomes and particularly be between regions of similar function. Now, this drive towards parsimonious wiring and a tendency of keeping the wiring as low as possible has been argued to be an important driving factor for the formation of specialized brain systems in the human brain. A second common theme that we emerge across all connectomes, again from the worm to the rat, from the pigeon to the monkey, is that strong investments are also made in features that might bring topological integration, in features that bring global connectivity across the system. All connectomes that we studied displayed features like hubs and the Rich Club, who despite their relatively high cost, may play an important role in keeping the overall brain network globally connected and facilitate global integration. Now, in a recent study, we further investigated this balance in terms of comparing the amount of gray and white matter volume of the brains of a wide range of primate species. Now, this is work done by a former student in my lab, Dirk-Jan Dash. Now, gray matter is the tissue, of course, where the neurons of the brain are located, and with that, their ultra-short range connections. And the white matter could be seen as this substrate for being the infrastructure for the long-range global connectivity across the brain. Now, it is well known that total brain volume is directly related to brain size. Basically, the larger the animal, the larger the brain. And this is, of course, very easy to see and understand. Now, in this study, we now examined further the scaling between, in particular, the gray and the white matter volume across species. And as expected, a larger brain was related to a larger cortical surface area, and more cortical surface area was in turn also related to more white matter. Now, interestingly, cortical gray matter volume was, or seemed to be outpaced by the white matter volume, with the coefficient being larger than 1 across the range of primates, suggesting that the relative proportion of white matter is increasing when brains get bigger. And indeed, if we look at the hard numbers, in humans an estimated 41% of brain volume is spent on white matter, while this is 40% in the chimpanzees and 27% in the macaques. Now, these findings are actually very similar, and in particular the estimate of the coefficient is very similar to earlier observations of Heuvel and Insel. Now the question, of course, is this increase in white matter enough to keep the brain connected? However, what we should be noted is that in an expanding network arguably requires the addition of many more connections to maintain global connectivity, which may quickly outpace the amount of space and biological cost that could be afforded by this system. So we further examined this by particularly looking at the corpus callosum, which is one of the largest and most prominent tracts in the brain, connecting the left and right hemisphere. Now we picked the corpus callosum as its volume can be simply estimated by just making just a sagittal slice and looking at the surface area or the area of the corpus callosum in that slice. Now interestingly, what we observed, again confirming these earlier observations of Heuvel and Insel, was that larger brains have a relatively smaller corpus callosum, indicative of long-range connectivity to decrease in larger brains. So we next compared graph-theoretical connectome properties. Examining the relationship between clustering and brain volume confirmed a stronger focus on local connectivity in larger brains. And furthermore, we also found that larger brains also have a longer path length, or therefore a relatively smaller or lower level of global connectivity. Now, Heuvel and Insel earlier proposed that this increase in local connectivity may catalyze the formation of more specialization in brain systems. So we further looked into that. And indeed, when we looked at the graph-theoretical metric of asymmetry between the left and right hemisphere, basically comparing the level of overlap and differences across the wiring architecture in the left and right hemispheric connectivity systems, we observed a higher level of network asymmetry in larger brains, with the human brain in particular being obviously the largest brain of the primates that we studied to display the largest left-right hemispheric differences. Now these findings are of course in line with early comparative work comparing in particular the arcuata fasciculus between human, chimpanzee and macaque. Now the arcuata fasciculus is a particularly interesting tract because it is well known to be crucial for the language system. It connects among other areas Broca and Wernicke. Now combined, these findings may suggest that connectome alterations related to an expanding brain may have given rise to preference of an increase in particular intrahemispheric local connectivity, potentially playing a role in the specialization of specific brain regions. So inspired by this work, I reached out to Professor Heuvel a couple of years ago, and we started on this amazing journey together of examining the connectome of the chimpanzee and the human brain and comparing them. Over the years, we developed several methodologies and methods to reconstruct the chimpanzee connectome, and one of the aspects we particularly spent a high amount of time on was the question of how can we fairly compare the connectomes between these two species. For this, we digitalized old cytoarchitectonic atlases from pioneers like Brodmann and Bonin and Bailey that identified homologous regions across these two species and, together with other great apes, to be able to match the nodes of the network. Now, this work was done by a former postdoc in my lab, Lianne Scholtens, who is a brilliant comparative biologist. Now, all of this work led to that we could do a fair comparison between the chimpanzee and human connectome. And what we found is just like we share a fair amount of our DNA, we also share a large amount of the circuitry between the two species. Now, we were, of course, particularly interested in also zooming in potential small deviations. So Dirk-Jan Adersch did some amazing work on this, and we examined these small deviations in the connectome in humans compared to the chimpanzees, and found that they were mostly focused around multimodal areas, brain regions that we know to be crucially important for higher order cognition. Now interestingly, in a recent comparative study, together with Luca Gocci and James Pang, we further examined the integration capacity of the human and chimpanzee connectome. And for this we use advanced computational models now to estimate brain functional dynamics. And we found that the human brain has a dynamic range distribution supporting faster integration between brain regions. Now this further inspired us to compare the cognitive systems between the two species. So in a follow-up, we performed a CWAS study in humans, first identifying those connections in that circuitry that relates to individual variation in cognitive abilities among humans. And next, together with Bill Hopkins of the National Chimpanzee Brain Resource, we found that the same connections could also explain the variation in cognitive abilities in the chimpanzees. So these findings thus suggest that cognitive systems might actually be shared by our common ancestor, and thus arguably are much older than the human lineage. Now furthermore, we also find that while some systems may have become more connected in humans, other systems might actually have become a relatively focus of adaptation more in the chimpanzee brain. So taken together, comparative neuroimaging studies allow for an examination of how the human brain may have developed and how adaptations to the brain may have led to the development of specific type of brain functionalities. Now as mentioned, we do of course have to realize that humans did not evolve from chimpanzees, and the chimpanzee-human comparison discusses adaptations over a relatively long period of time. So what about more recent evolutionary adaptations? Now, as we mentioned already in the beginning, fossil records are here the prime method of choice, but they have their own limitations. In particular, that they cannot capture soft traits. They can only capture things like cranial capacity and gross features of brain organization. Now, one alternative way might be to look at our genes. Now, the human genome may be considered as this timeline or a photo book of memories of human history. Mapping the genetic timeline of when specific single nucleotide polymorphisms, SNPs, associated with modern day phenotypes, when they first emerged in our genome, may be a window to explore the evolutionary tempo of characteristic features of humans during hominin evolution. Now, in a recent study, a student in my lab, Ilan Lebedinsky, did indeed do this. So for this, we used a unique database of the Human Dating Genome Project, which provide estimations of when certain SNPs emerged in our genome. So Ilan took a wide range of GWASs from the Human GWAS Atlas, and we looked at when all of these SNPs together emerged in the genome. Now, interestingly, when we looked at this accumulated timeline, we found basically two bumps, peaks that seem to be strongly related to well-known milestones in the Homo lineage. So next, we split all of these SNPs according to specific traits, starting from gross categories, moving step by step to more fine-grained traits. And we found that the class of traits related to psychiatric disorders appear to be among the youngest in the human genome. Furthermore, cross-referencing this with gene expression data from the Allen Human Brain Atlas, we found that youngest genes seem to be more expressed or overly expressed in the pars triangularis and temporal cortex, areas which we well known to play an important, crucial role in the language system. Now, as you can imagine, we are very excited about this new method, and we are now looking into options to further combine these two SNPs related to brain volume and function. So taken together, I hope I have highlighted several examples of how comparative analysis and comparative neuroimaging and connectomics can provide us with new insights into how human unique and species common brain systems may have been formed, and how selective and environmental pressures may have played an important role in shaping the brain of humans and, in that sense, brain function. Thank you very much for your attention.
All right, so unfortunately Martijn can't be here, so we won't have questions. But I will encourage you to use the app and note down your questions for our subsequent speakers, so then we can ask them. We'll probably aim to do one question per speaker, and then whatever time we have left, we can open up to a panel discussion.